Hello everyone. Hello everyone. Thank you so much for joining. And we're going to talk a little bit about the Rhode Island NGSA today. And we're excited to get into it. And we're going to start introducing ourselves now. Emily.
All right. Thank you, Julia. So my name is Emily Miller. My pronouns are she, her. I was a high school chemistry teacher for 10 years. During that time, I taught in our alternative learning program. I taught regular chem and honors chem. So I had an opportunity to try a lot of different strategies with a lot of different kids. During my time, I was an NGSS pioneer and advocate in my science department, and I was also a PLC lead. So I got to lead a lot of data-driven dialogue and data chats. And now I get the absolute pleasure of partnering with districts to help them implement InnerOrbit. And I kind of like, well, no, I just do. I like savory more than sweet. This isn't to say like I'm a sweet hater. Like I love chocolate, but I will order an appetizer before and a dessert like any day of the week if I had to pick. Julia, do you want to introduce yourself?
Yeah, I don't necessarily agree with that, but good for you. So my name is Julia Mota. I was a teacher and tutor for elementary students in the Dominican Republic for quite a while. But now I am actually supporting teachers and districts with their NGSS implementation. And I've been doing that for the past 3 years. I'm grateful to be InnerOrbit's district engagement manager. So I'm able to partner and provide guidance to schools and districts implementing assessment in their NGSS instruction. I also think that matcha is better than coffee. So I don't know how you feel about that. I know there are some coffee lovers, but yeah, I don't love it. So
Yeah, I don't know if I agree with that either. I mean, matcha's fine, but like coffee is life for sure. So for our objective here today, we get the opportunity to analyze some release test items and dive into resources and strategies to help practice and prepare for the Rhode Island Next Generation Science Assessment. We'll be here for about 30 minutes today, during which we're going to start off actually by looking at that release test item, and we're going to do a little bit of analysis with it. And then we're going to do some practicing with sense making. And to do that, we're going to talk about how we can use phenomena throughout a unit. We're going to talk about how we can scaffold back a grade level if necessary. And then from there, talk about how we can scaffold up to 3D questions, looking at some InnerOrbit items. While we're doing this webinar, let's get started. One thing we wanted to do for you all really quick is just talk about what InnerOrbit is, if you are not familiar. So the first thing InnerOrbit is phenomena. So in order to truly sense make, in order to truly be a part of the NGSS, students need to be able to make sense of novel phenomena. So our site is filled with a lot of novel, new, interesting situations that students need to make sense of and apply their skills and learning to. They do that through answering questions. So our site has over 10,000 different questions that are all aligned to the NGSS. And one thing that we do really well is these assessment items are actually scaffolded. And Julia is going to talk a little bit more about that later. But what we do is we slowly build students to be able to answer those really rigorous three-dimensional questions by offering 1D and 2D questions leading up to those rigorous items. Another thing that we are is reports and data. And not going to lie, this is probably my favorite part about InnerOrbit. So as students are taking the assessments, you actually get this really detailed feedback as a result. Not only do you see how students are doing on the assessments, but you see how they're doing specifically on the SEPs and the CCCs and the DCIs. So you can take like a really rigorous three-dimensional question and actually pull apart each dimension with this report and see which aspects students might need some more support with. The last thing that InnerOrbit is professional learning. So just like this webinar that we're doing here, we like to offer a variety of research and context-based professional learnings. We can talk about how to model engaging classroom strategies. We love receiving feedback and driving our professional learning around that as well. But we want to be able to educate as much as we want to offer our assessment platform to people as well. If InnerOrbit is something that you think you might be interested in, Julia just dropped a link in the chat where you could book some time to meet with her, and she would be more than happy to talk to you about how InnerOrbit could potentially look in your school or in your district. So now that we talked about InnerOrbit and you know a little bit about us and what we do, let's take some time to actually look at that Rhode Island assessment and talk about sense making. So this assessment item that we're looking at from the Rhode Island NGSA is an 8th grade practice item. And it reads, flies with bar-eyed phenotypes cannot see as well as those with wild type phenotypes. The genotypes and phenotypes of 3 flies are shown in figure 1. Organize the statements that complete the chain of events, explaining how the bar mutation reduces a fly's eyesight. So we've got this stream of phrases here. So phrase one, then an arrow, and then another and another. So students would need to grab those statements and then place them in those varying 3 spots there using the information given. So what we would like you all to answer right now is, if in the chat, could you please share, do you think students are applying their understanding of DNA and proteins to make sense of this phenomenon? I would agree that yes, they are applying their understanding of proteins and DNA in order to make sense of that phenomena. So let's just take a minute and talk about what we actually mean by sense making. So representation is something that you're probably familiar with. This is where students are communicating ideas that they've previously learned. So essentially being able to like regurgitate information that they've been told. This is an important part of learning. It's really good for diagnostic and formative assessments to really know what your students know prior to moving forward and teaching them additional content. While it's important, it's insufficient evidence of proficiency. We don't really know if students understand DNA and proteins if they are simply just recalling information that they've learned before. Sense making is the tool that we can actually use in order to see if students actually have gained that proficiency. This is because they need to explain why or how while they try to make sense of a novel phenomena. This is a great summative assessment opportunity because students need to apply what they've already learned to a new situation. And this is ultimately the goal of both the Rhode Island NGSA and the NGSS as a whole, to be able to really demonstrate the mastery of that standard by making sense of something new and novel. So thinking back to that fly eye question, this was really looking at the performance expectation MS-LS31. So being able to develop and use a model to describe why structural changes to genes, mutations, located on chromosomes may affect proteins and may result in harmful, beneficial, or neutral effects to the structure and function of the organism. So that would be like your standard. And this standard has 3 different dimensions that feed into it. The first is the DCI, or the disciplinary core idea. And this is the content. This is what students need to know. So students need to know that genes are located in the chromosome of cells, and that this kind of maps out the DNA structure and moves forward with producing proteins and other things of that nature. I was a chemistry teacher. Forgive me if I botched that a little bit. The science and engineering practice, this is what students actually need to physically do. This is like the skills that they actually learn. In this case, being able to develop and use a model. So students looking at those images of the flies and then being able to drag and drop those sentences into a series of arrows is them being able to use a model to describe the phenomena that they're seeing with the fly eyes. And then the cross-cutting concept is a way for students to kind of frame or think about the phenomena. In this case, the CCC is structure and function. So being able to look at complex microscopic structures and systems and see how all of those working parts can come together to affect other various parts of the organism. So bringing all of these dimensions together, it makes up that performance expectation of MS-LS31. And if you notice, all 3 of those dimensions are taking a part in answering that fly eye question. So what we need to be able to do is we need to be able to support students, not just with content knowledge, but we need to be able to support them with the SEPs, so the skills that they need to do, and the CCCs, being able to help them frame and look at a situation through a particular lens. And Julia is going to talk to us a little bit more about how we can do that.
Thank you so much, Emily. Now we are going to talk about how to use phenomena for test prep and how we can actually gather data around all that so that our students get used to transferring that knowledge to novel phenomena. Now, going back to our standard MS-LS31, and just to recap, it is asking students to develop a model to describe why structural changes, mutations located on chromosomes may affect proteins and may result in harmful, beneficial, or neutral effects to the structure and function of the organism. So how can we give students opportunities to make sense of phenomena throughout this entire lesson? This is a roadmap that we've built of phenomena on the InnerOrbit website that we would use throughout this lesson. And so in the very beginning, maybe we could start with this phenomena discussing, why is this hedgehog white? And since we're still in the beginning of the unit, maybe we consider a lower stakes discussion, like a whole class walkthrough. And this is all about how albinism is a genetic condition caused by mutations that affect the product of melanin, which is the pigment responsible for skin, hair, and eye color. And we would include discussion questions that target the cross-cutting concept and the SCP for the whole class to answer together. So we would scaffold this in different activities, and the first one would be a whole class walkthrough and discussion. Moving on to the middle of the lesson, we could talk about the reason why people from Tibet can breathe so easily at high altitudes. And here we could consider separating the class in groups to answer questions collaboratively in a printed and scaffolded group phenomena exploration. So students would discuss in this case that certain populations living at high altitudes, like those in the Tibet, have developed genetic adaptations that help them cope with lower oxygen levels. And in this stage, students are answering rigorous questions that target the DCIs, the cross-cutting concepts, and the SCPs, but with scaffolding from the teacher and support from their group. Now, lastly, we could assess students by building a short online assessment to be administered individually. And we would, in this case, cover the reason why some people are able to roll their tongues while others can't. So this is something when I was little, when I took this class, I was like, I know I can do this. I couldn't. So in this case, students will grapple individually with the fact that the ability to roll the tongue is associated with genetic variation in a specific gene. And this trait is neutral and has no clear impact on an individual's fitness. But in this final part of the unit, students would be able to engage with rigorous sense-making questions individually with no scaffold. So it is important that even though you can provide scaffolds at this point of the unit, removing those is as important for building student independence and confidence when answering these questions. Now, I do want to mention that all these phenomena you can find on InnerOrbit and even more. So per PE, we have about 4 to 6 different phenomena for you to use throughout a unit. But let's say maybe our students aren't there yet, and we're starting at a level where they need even more phenomena, and they need even lower level phenomena to access these rigorous things. We've got something for that. So in this case, what we would recommend in this situation is for us to go from the grade level standard to a prior grade level standard. And in this case, we're going back a whole grade level to this 3rd grade standard, where students have to analyze and interpret data to provide evidence that plants and animals have traits inherited from their parents, and the variation of these traits exist in a group of similar organisms. And again, even though we're looking at this middle school standard, this definitely applies, because I know we have some biology teachers in the chat. So this definitely applies to biology too. I know we have some middle school 8th grade and 7th grade too, so yay. But this also applies. We would go from high school, we would go from a prior grade level, and we would go to middle school. Now, we would do the exact same thing here. We would use multiple phenomena to guide students throughout this lesson. So maybe we start discussing how tigers get their stripes. And this is all about how many characteristics of organisms are inherited from their parents, and how different organisms actually vary in how they look and function because they have different genetic inherited information. And then we could go over the reason why the same type of flower can look so different. And it's all about the fact that tulips can be crossed to create a mix of flowers with some similar and some different traits. And again, all of these and even more can be found on InnerOrbit. So if you're looking for phenomena to include in your lessons, InnerOrbit is a great tool for you to look at. Now, to sum up how we would use phenomena throughout test prep and why this is important. First of all, practice makes perfect. So we should be using phenomena throughout all units, throughout all lessons, not only during test prep to prepare our students for a specific test, but using also different phenomena provides even more opportunities for students to model and practice transferring knowledge and sense making. And if we're able to give them multiple opportunities to do so, we're able to actually help them perfect that skill. And if students are struggling with phenomena targeting their own grade level band, we can always lower the stakes with a prior grade level phenomena to give further sense-making opportunities and identify possible gaps in knowledge or in skills. And that pretty much sums it up about using phenomena throughout a unit or a lesson. Now, we've actually talked about scaffolding with activities. Now, let's talk about how we can move and how we can scaffold from representation to sense-making with different types of questions. So our goal at the end of the day is to get students to that rigorous performance where they can use their knowledge through the lens of the cross-cutting concept and the SCP to explain why or how a phenomenon happens. And that's kind of like what true mastery looks like. And it is what students need to be prepared for these assessments. But we can't expect to get them there overnight. And that's when scaffolding comes in. So how do we support them in building up to this level? First, we're going to identify the invisible steps that students are going to take. And if we're expecting to go from recall to applying knowledge across dimensions, we have to be intentional about breaking that journey into manageable steps. And second, we also need to gather data at each step. So not just at the end, not just with a summative assessment, but we actually need to gather data throughout the school year to understand where students are struggling and adjust our support accordingly. And by focusing on these steps, we're not just preparing students for an assessment, but we're also helping them develop the critical thinking and the reasoning skills that would help them far beyond the classroom. And that's what we expect them to do with sense making. Now, again, how do we bridge that gap? Let's do this gap from DCI recall to the three-dimensional questions. Let's look at it with an example. And in this case, this is all about how X-rays expose us to radiation, which in high doses can cause mutations, changes in our DNA. And sometimes these mutations may have no effect, but in other cases, they can lead to serious consequences like cancer or inherited genetic changes. So on this slide, we see a few key ideas. The first one is how radiation can cause DNA strand breaks, leading to those mutations that we talked about, how some mutations affect only the individual while others can be passed on. And we also see an example of how radiation can break and rearrange chromosomes, leading to genetic changes. So our end goal is for students to be able to answer questions like this, right? So this questions requires students to engage in three-dimensional sense making by pulling together multiple scientific dimensions, right? So we have the science engineering practices, the DCIs, and the cross-cutting concept, which is structure and function. And in this case, this level of complexity is exactly what we see on the Rhode Island NGSA. So how do we get there? With two-dimensional questions. So since we said we can expect students to jump straight from representation to sense making overnight, these is where two-dimensional questions come in. And these act as a bridge between simple recall and full sense making. So some two-dimensional questions combine the DCIs and the cross-cutting concepts, asking students to explain why or how scientific knowledge applies through a specific lens. And some others combine the DCI and the SEP, shifting students from recall to application by having them engage in scientific practices instead of just memorizing facts. So incorporating more 2D questions in our instruction helps students build the skills that they need to fully engage in sense making. So it's a stepping stone at the end of the day that makes the shift to mastery to these three-dimensional questions feel much more achievable. Now let's look at how we would build up to those questions, to that sense making question that we just saw with different types of questioning. So the first one is a great example of a DCI only question. This is representation. Again, this plays a good role in assessing whether students are ready to start making sense of something novel. So because before they can explain, they can model, they can analyze the phenomena, they have to have a solid grasp of the foundational science idea. So in this question is specifically checking by focusing on purely the DCI, which is variation of traits, and it is assessing whether students can understand that mutations change the protein our body produces, and it also acts as a checkpoint. So students can answer these questions, check, then they have the necessary foundation to move on to more rigorous questions. Now, for a second type of question, we combine the DCI and the cross-cutting concept, intentionally leaving out the SEP. And as I mentioned before, this type of question bridges the gap between simple recall and full three-dimensional sense-making by ensuring that students can recognize, in this case, how DNA mutations affect protein production and traits, and they use structure and function as a lens to explain why altering DNA leads to new proteins and potentially different traits. Then we do the same thing with our science and engineering practice. So for this third type of question, it is deliberately combining the DCI and the SCP while living out the cross-cutting concept. This question is actually isolating the SCP for us to understand where the gaps are and adjust instruction accordingly. So if they were able to meet the performance maybe for other questions, if they were able to answer the DCI only question, if they were able to answer the cross-cutting concept question, but they struggle with this one and the three-dimensional question, then what they may need support is specifically in developing and using models. So again, we're trying to gather data and understand how we can not only scaffold student thinking, but also gather those useful insights that will help us make those adjusted and adjust and make informed instructional decisions towards the end. And then finally, looping it back to our three-dimensional question, this question is a true sense-making task because it requires students to engage with all three dimensions. And it's the kind of question that best prepares students for the Rhode Island NGSA. Instead of just identifying facts, in this case, they have to recognize how DNA mutations affect protein production and traits. They have to identify which of the classmates modeled correctly how mutations affect organisms on a molecular level. And they also have to use structure and function as a lens to explain why altering DNA leads to new proteins and potentially different traits. So again, by scaffolding students through representation, two-dimensional thinking, and then three-dimensional sense-making, we ensure they are truly developing the skills that they need, not just for assessment, but for real scientific problem solving. And this is where we want students to be. By breaking down this journey, we make it a little bit more accessible, and we can support every learner in getting to this level of thinking. So just as a recap, the types of questions that we went over: first, we start with DCI-only questions, just representing their knowledge, just recalling facts. Then we combine the DCI and the science engineering practice, the DCI and the cross-cutting concept. And finally, we build up to those three-dimensional sense-making questions, which at the end of the day, our goal is for students to be able to independently answer these without any scaffolding, without any help from their teachers or from their colleagues, because these are the types of questions, not only that they're going to find on the Rhode Island NGSA, but also those questions that meet the level of rigor of the performance expectation. And I wanted to share very quickly some sample data. So this is a data set that from a district that has given assessments across all school year. And as you can see, they have gathered data throughout different dimensions, and they are able to see and understand where specifically they need to hone in on and where their strategies have been working the best. Now, over to Emily to wrap us up. We've done so much in just under 30 minutes.
Yes, we have. And thank you so much, Julia, for all those awesome explanations for how all of that is broken down. So here's a couple of next steps for y'all. First of all, Julia is going to drop another link in the chat here. You can click on that in order to book a meeting with her on her calendar, and she would love to chat with you about InnerOrbit and potentially starting a free pilot this school year. Our free pilot program usually lasts 1 to 2 months, and you would have full access to our site and the ability to actually administer our questions and assessments to your students. And this would be an awesome way to try some of these prep strategies that we talked about to get ready for that Rhode Island science test. You can also follow us on social media. Our YouTube channel is actually where we post all of these webinars in addition to other resources as well. So at InnerOrbit NGSS is where you can find us on YouTube. And if you have any questions at any point where you want to discuss InnerOrbit or anything else we can do to help you, you can always email us at inquiries@innerorbit.com. Julie and I will hang around for a couple of minutes just in case any of y'all have any questions that we can help answer. But otherwise, thank you so much for joining us today. It was great to have you all here. And if we're here, if you need us, if you've got any questions or want to learn more. Thank you so much.